document, if we suspect we have misspelled a word, we can use the find function to highlight the error and correct it or delete it. Within our DNA, the function is taken on by a system called CRISPR-Cas9. CRISPR is short for cluster regularly interspersed short palindromic repeats. CRISPR consists of two components, the Cas9 protein that can cut DNA and a guide RNA that can recognize the sequence of DNA to be edited to use. CRISPR-Cas9 scientists first identified the sequence of the human genome that's causing the health problem. Then they create a specific guide RNA to recognize the particular stress of A's, T's, G's, and C's in the DNA. The guide RNA is attached to the DNA cutting enzyme Cas9, and then this complex is introduced to the target cells. It locates the target vector sequence and cuts the DNA at the point. Scientists can then edit the existing genome by either modifying, deleting, or inserting new sequences. It effectively makes CRISPR-Cas9 a cut and paste tool for DNA editing.
Now it's my turn to explain about the example of CRISPR-Cas9 in cancer treatment. So the CRISPR-Cas9 enhancing T cells. Firstly, I will explain about what is CAR T cells. CAR T cells have a full name, which is chimeric antigen receptor T cells, and it's genetically engineered T cells to target cancer antigen. CRISPR-Cas9 improving CAR T cell function in three terms, which are enhancing, overcoming, and silencing. The CRISPR-Cas9 enhancing specificity of T cells to target cancer cells, and overcoming T cells exhaustion through cytokine regulation, and lastly, CRISPR-Cas9 silencing immune suppressive pathway. And the following diagram shows the difference between the normal T cells and CAR T cells. For the normal T cells, TCRs interact with antigen process and presented by MHC1 on the surface of tumor cells. This interaction requires the antigen to properly process and bind to MHC, which limits the range of target. For the CAR T cell, it is genetically engineered to express chimeric antigen receptor that contain three structures, which I have mentioned in the beside yellow box. CARs directly recognize and bind to antigen expressed on the surface of tumor cells, bypassing the need for antigen processing or MHC presentation, which allow it to target a broader range of tumor cells. And this slide shows the CAR T cells therapy step to treat cancer cells. If you are interested, you can pause and watch it. Now we come to the clinical result about the treatment refractory metastatic liposarcoma. There is a big reduction of the tumor size, and this could absorb by our naked eyes, so we can conclude that the CAR T cells therapy is a powerful tool in cancer treatment. That's all from me. Thank you.
Now allow me to talk about the ethical consideration of CRISPR-Cas9 application. First of all, it is vital to consider the biosafety of germline genome editing. This is because the high incidence of off-target mutation and genetic mosaicism raise serious questions about the safety of using CRISPR-Cas9 for germline editing, especially the potential to pass on unintended genetic changes to future generations. Hence, in 2015, the International Summit on Human Gene Editing called for a global moratorium on germline editing for reproductive purposes, recommending further research and discussion before proceeding with clinical application. Next, there are ethical issues raised about the equal access of CRISPR-Cas9 technology used among the different layers of society. This is because this technology is expensive and only the wealthy family could afford to choose to genetically enhance their children. Those without access to this enhancement could face disadvantages in society, leading to greater economics and its social polarization over generation. On the other hand, from the perspective of global level, wealthier countries are more likely to develop and have early access to CRISPR-based technologies. In contrast, lower-income countries may struggle to access the technology. This will cause only certain populations are able to benefit from life-saving or life-enhancing genetic modification. Next, one of the biggest dilemma of the application of CRISPR-Cas9 is to obtain informed consent. Individuals or their legal guardians should understand the risks, benefits, and long-term consequences of the application of CRISPR-Cas9 before giving consent to proceed. Last but not least, CRISPR-Cas9 may provoke ecological imbalance. Since gene drift will proceed in a population, possible off-target mutation will continue in each generation. The possibility of regulated gene transfer to other species may result in the transmission of negative characteristics to the associated organism.
I will explain how this CRISPR-Cas9 technology is making strides in human health and medicine, as well as its profound impact on agriculture development. For people with diabetes, CRISPR-Cas9 provides hope by creating insulin-producing cells from a patient's own cells, reducing the need for transplants and improving disease management. Next is malaria. Malaria kills hundreds of thousands every year. CRISPR-Cas9 is being used to modify mosquitoes so they can't spread this disease. Scientists use gene drive to make mosquitoes infertile or produce only males, reducing their numbers and preventing malaria transmission. Antibiotic resistance is another growing problem, and CRISPR-Cas9 offers a solution. It can precisely target and remove harmful bacteria, even resistant strains, while keeping good bacteria safe. It is a smarter, more effective way to fight infection. In agriculture, CRISPR is improving crop by making plants grow more food, resist pests, and survive harsh weather. For example, scientists have developed rice resistant to diseases and corn that tolerate drought. CRISPR also helps control pests by reducing harmful insects without using chemicals, promoting sustainable farming. In conclusion, CRISPR-Cas9 is a revolutionary tool that can transform healthcare and agriculture by curing diseases, combating antibiotic resistance, and improving crops and livestock. However, it must be used responsibly, with ethical considerations and regulations to ensure its benefits are shared fairly and risks are minimized. By advancing research and establishing thoughtful regulations, we can harness CRISPR-Cas9 to create a better future for everyone. That's all from our group. Thank you for your time and attention.